Toxo loves the brain, so I think we're a ways away from it, but I think there are valuable things we could learn if we could grow them in, neuron cells or neuron plus astrocyte models that might tell us a lot about Toxoplasma.
Hello, everyone, and welcome back to People, Parasites, and Plagues, a podcast aimed at delivering information about the fascinating pathogens among us from the scientists who study them. I am Kim Klanowski.
And I am David Peterson, your host for today's episode. There's a 1 in 8 chance you are infected with the parasite studied by today's guest. And no, ivermectin won't help.
Our guest today is Dr. Robyn Kent, Assistant Professor in the Department of Microbiology and Immunology at the University of Oklahoma College of Medicine. Robyn, welcome to the podcast.
Thank you for having me. It's a pleasure.
So our first go-to question is about your pathway to science. So what brought you from, I don't remember where you grew up. You told us yesterday.
Somerset, England.
Somerset.
Yes.
What brought you from Somerset, England to Oklahoma and assistant professorship?
So I started off growing up on a farm, and I really loved being around animals. And so I thought that's what I wanted to be when I grew up. And then I very quickly realized when I was about 6 or 7 that I didn't like being dirty and I didn't like manual labor. And so farming didn't really seem like the right choice. So I kind of became interested in being a vet, which is a next logical step. And then I quickly realized that also wasn't for me. Agricultural veterinary work is a great profession, but involves a lot of putting your hands in places I didn't really want to put my hands. And when people have sick pets, they're kind of sad, and I didn't really like that either. So I turned from my local environment to the glamorous world of television and thought I wanted to be a forensic scientist. And then I realized there's a lot of paperwork and not a lot of glamour. And I had some great professors when I was training or trying to train to be a forensic scientist. And they opened my eyes to a world I'd never even thought of, which was symbiotic interactions and parasites. And that was it. It took me a few stumbles to get there. But once I had that microscopic life open to me, I was hooked. And so that's where it came from.
And I do want to say that I was a postdoc at the same time that Andy Waters and Gary Ward, your two mentors, were at NIH. So a shout out to both Andy and Gary.
That's such a coincidence. I had no idea they even overlapped, actually. Neither one of them have ever told me that.
I think they did. I know they overlap with me.
Mm-hmm.
So maybe not with each other.
Could be.
But that's okay. So Robyn, what are the fundamental questions your research addresses?
I guess the fundamentals are, how does a parasite persist? What does it need to do? What can it get away with not doing? How does it stay hidden? How does it balance that? And how, when the parasite gets these cool signals, does it respond so quickly to do so many different things? Because that's one of the great things about Toxoplasma, is when it gets a signal, it rapidly does whatever it needs to do to survive and keep going in whatever direction it needs to. And I think that's pretty cool.
So Toxoplasma has one of the more complicated life cycles. So I think for today, we'll kind of skip the life cycle in the cat, which is the definitive host, because then we can just deal with the fast dividing forms, the tachyzoites, and the slow dividing forms, the bradyzoites. Actually, I guess some people used to think that the Bradys didn't divide.
Yes, that is the age-old dogma that I'm trying to break down. Many people in the field did just think once you had a chronic infection, it just sat there, not doing anything interesting, just kind of hanging out, waiting for your immune system to crash or for you to get eaten by your cat or by some other animal so that it could transmit. But some really cool work has come out in the last, 10, 15 years that has questioned that. And I've always thought, if you just go to sleep, how are you ever going to wake up quickly if you're too lazy, if you're not doing anything inside that cyst? And so when that work started coming out, I was like, I can build on this. I can make my contribution here. I've never thought they were completely dormant. And the more I talk to people, the more I think everybody suspected that wasn't quite the case. And so I think that they're replicating. I think that they are leaving home and making their own new cysts. And I think sometimes they're doing division of the whole cyst and taking it with them, which I think is pretty cool.
So do you think that the need to kind of divide and maybe make a new cyst is something that's intrinsic to the cyst itself, or is there an environmental trigger you think that's kind of driving this process?
I think yes. Yes to both of them.
Okay.
I think that, like I think of me living in my house. If my house is crumbling around me, I'm going to get the heck out and maybe get a new one.
Right.
And so I think there are those within cyst signals that say, this is not the right environment for you. You need to get out. But I also think there is signals that say, I'm old or I don't have enough friends or I have too many friends here. And so there's parasite to parasite signals that say, the cyst is fine. The house is fine, but you need to leave, go off on your own. So I think there are both signals from inside and from outside that tell them what to do. And I think we're so at the beginning of understanding it that we'll find a lot of different cues, which is nice to have those options.
Yeah. And maybe we should talk for our listeners who don't understand. The cyst is not a single parasite.
No, so they go into a cell as one parasite and then they start replicating. And so every time a Toxoplasma replicates, it goes from 1 to 2, 2 to 4, 4 to 8. And whilst they're doing that, they're also building a wall around themselves. So they're considered clonal because they came from that one parasite, but a cyst contains hundreds, if not thousands, of individual parasites inside itself. And some of these can be huge. 100 microns in diameter in your brain, and the ones in your muscle cells they kind of look like hot dogs, so they're really long on one axis. I think the longest one we've seen in a mouse is 300 microns long, and it contained several thousand parasites.
So the cysts aren't, and this is a parasitology question, sorry. I thought the cysts were always round.
In the brain they are.
Oh.
So they're big spheres in your brain. But I think as your muscles contract, they force them, and they're spherical. Well, they're not spherical. They're circles on one axis, always. But then they become like hot dogs, elongated along the axis of your long muscles, which I just think is cool. And I think that the kind of force that you get from muscles contracting and stretching is what causes this to happen. And it's kind of cool because people don't look at the muscle ones very often. And that's why most people think that they're these spheres. But if you just look in the muscles, they're different.
So there's a reason why I guess they focus on the ones in the brain, and maybe you can tell us something about that.
Everybody needs a brain to be alive. That's one of the reasons. So if people have Toxoplasma, you have a large-ish number of cysts in your brain. And if your immune system becomes suppressed, then they wake up. And anything that wakes up in your brain and starts replicating and kills cells and causes all this damage is very bad for you. And so the biggest disease manifestation for Toxoplasma is encephalitis in your brain. And so if that's the part that kills people, that's the part we tend to focus on. It's also the easier part. I wouldn't say Toxoplasma research is easy, but getting the cysts out of the brain pretty much is. We don't have isolation methods for muscles. Everything we've tried, if you break the muscle, you break the cyst. And then if you can't get it out, how do you study it?
That's tough.
So yeah, I torture each of my graduate students. They get 6 months and they have to try and find a method to get them out. And so my first graduate student, she tried maybe 15 enzymatic methods, and none of them worked. And so she was like, I want to try this next. And I was like, nope, you've paid your dues. The next person can try it. I don't want you to bang your head against a wall forever.
So is that because of the muscle cells itself, or is it because of the cyst, the type of what's surrounding?
I know, but I keep saying both for everything now. I'm sorry. It's so muscle cells are hard to get apart. They like to be in these long fibers and bundles. And so breaking them apart is challenging. And some enzymes do it by breaking down the connections between those muscle fibers. And it just so happens that the things that can break down those connections between cells also poke holes in the cyst wall. And so it seemed like a good idea at the time. And then, when you think about it in hindsight, as we always do, it was maybe an obvious one that it was also going to poke those holes. And so we're thinking of kind of using a grinder next to try and mechanically break everything apart.
I think I've heard that organoids are the answer to everything now.
Yeah, they're great. We do think about organoids, or we think about complex cell structures that might allow us to grow the parasites. The brain is the hardest organoid to make. I think it's the one that we haven't really done as well with as the gut ones are fantastic, right? We all can accept those. But Toxo loves the brain, so I think we're a ways away from it. But I think there are valuable things we could learn if we could grow them in, neuron cells or neuron plus astrocyte models that might tell us a lot about Toxoplasma.
Because right now, that's the protocol that you're relying on, is infecting animals and then pulling them out. Nothing really is happening in vitro.
Yeah, so that's one of the, bugbears I have with everything is our current model for the bradyzoite or the chronic stage is that we just really stress them out. And it causes them to start making these cysts. But the work that I've done since I started the lab has shown that, they look like a cyst and they express these early markers, just don't go all the way. And that's great if you're asking questions about how they make cysts, but I want to know how they stay a cyst. That's the thing that I think is really cool, is if a person gets toxo when they're 10, they'll still have it when they're 90. And that's really, really weird and really cool.
Do you think that the signals that are used to keep dormancy or start replication or come out of dormancy is different between cysts that are in the two different cell types, in the brain or in the muscle? Sorry.
No, I love it. I really think that my brain is quite different to my muscles. My neurons are. And so, I think they're really different in every way. If you live in a brain, there's no immune cells trying to kill you, or there's very few. I think that neurons fire with electrical signals, and I think that muscles contract and try and crush you. And that if you are living in a muscle, maybe your shelf life is lower. Maybe replication is faster. Maybe it happens more, because you can only live 6 months rather than 5 years. And I think that it's going to be critical, if we want to get rid of Toxoplasma, to not just look in a flask and not just look in a brain, but look everywhere that Toxo can live. And that includes your heart and your legs and your eyeballs. So we should be looking at all of the places.
So there are parasites related to Toxo, like Sarcocystis species, that, at least in my mind, are more commonly found in muscle. But maybe it's because we don't have brain that often at the deli counter.
Oh, I hope not. But I don't know what you guys eat in Georgia. Maybe it's a state delicacy. No, so there's been lots of different studies on different animals and different meats. And actually, I think sheep, there is a lot of Toxoplasma, and it causes a huge problem with sheep farming, which is actually kind of how I connected with my dad, who was a sheep farmer. And so once I started doing Toxoplasma research, he became more interested in it again from the angle of how do farmers deal with Toxoplasma rather than how do people. And so consumption of undercooked lamb is a source of Toxoplasma infections. But I think lamb is much less popular in the US than it is in the UK and in European countries. So that could be one of the reasons that the United States has lower prevalence of Toxoplasma than other places. You don't eat enough delicious lamb.
Does it have an agricultural impact on the animals themselves?
Yeah, it's actually horrendous for spontaneous abortions in sheep. It's one of the reasons that farmers will just stop using a field, because it's been contaminated with the oocysts that come out of cats, and then the sheep eat it, and you have huge loss in your output, I guess. That's probably not the right way to say that. And there's actually a vaccine, Toxvax. I don't think it's used in the United States, but it's used across Europe. It kind of works.
That's true for a lot of parasite-directed vaccines. They kind of work.
Yeah. So I think, the agricultural impact is often overlooked as well.
So a lot of your work has involved, well, at least some of what you talked to us yesterday about, single-cell RNA-seq. I guess let's talk a little bit about what that does and specifically what you learn from it that you don't learn from regular RNA-seq, which basically tells you what a bunch of tissue is transcribing, which genes are being transcribed.
Yeah, so I think a lot of people use single-cell RNA sequencing on multicellular organisms, and then they can learn what is your B cell doing, what is your T cell doing, what is your red blood cell doing, or your other cells. But we use it to look at a population. I kind of like to think of it as the difference between what are people in Georgia doing versus what is Kim doing, and what is David doing, and what is Robyn doing. We're all doing something different. And if we look at the single cell level, we can answer what is parasite 1 doing versus what is the whole population as a whole doing. And I think that probably parasites and Toxoplasma aren't as different as people, but they are different from each other. And so you can ask questions in terms of what is something that is replicating doing versus something that isn't? What is something that is starved doing versus something that isn't? And how does the whole population differ at an individual level? And I think that's really, really powerful because you can detect very small changes or things that only happen for a short time, whereas the population kind of swamps that and you can just see what is happening as a whole. And for bradyzoites that I work on, it's been really good because we think there are subpopulations. And so in one cyst, there are 1,000 parasites, but 100 of them are doing something different to 900 of them. And if you just look at 1,000, you can't tell what that 100 is doing. And so it's kind of like, how do you say the detection of things that are just not seen within the whole population?
That's an interesting degree of specialization in a way, which we expect in multicellular organisms, but maybe is unusual somewhat in a single cell organism.
Yeah, so I think it's because of that complicated life cycle that you talked about at the beginning is Toxoplasma bradyzoites, the cysts, they have so much choice on what they can do. So if a mouse has Toxoplasma cysts in its brain, that mouse could be eaten by a cat. And if it's eaten by a cat, it gets signals that say, I think you should do the sexual cycle. I think this is the right choice for you. If the mouse is eaten by a pig, then it's not given those same signals. The thing that it's told to do is to go back to the actively replicating tachyzoite stage and to colonize that new host. And this is a bit of a ridiculous example in a mouse, but if the mouse becomes immune suppressed, because it's undergoing a cancer treatment or something, less likely with a mouse, then it's in the same host, but it is told that the immune system's gone away and it should go back to that fast form because it's been given the opportunity. And if you have subpopulations that are ready to do each of those things, you can survive no matter what life throws at you. And I think that's what Toxoplasma is really good at doing. And I think it does it because of the life cycle and because of the vast number of hosts that it can infect. It's not just people, it's everything. Every warm-blooded mammal can get toxo. And every warm-blooded mammal is slightly different. So its ability to adapt is what makes toxo really special.
Yeah, and it's interesting because you talked about it in terms of being clonal in the cyst, but then it's like what it's actually doing is different. It's kind of like if you cloned yourself a gazillion times, but then some trained to be a warrior and some trained to be, I don't know, a waitress.
Podcaster,
any of those things. So that's kind of, so is that, gosh, it kind of goes back to what are those triggers and what are those signals? And if are these certain clones sensing something different?
Yeah, that is totally unknown. And it is not just clones, it's clones that are in a box together, right? They didn't even get to go live in a different country to get some different experiences. They live within the same cyst and they come from a clonal start point. How the heck do they change so much from each other? What triggers it? What controls it? I just spoke to somebody today who said, do you think they are different at a genetic level because of SNPs? And I was like, well, I've never even thought about that before. The lowest I've gone before is epigenetics, which is something I never want to work on because I'm not an acronym person and epigenetics is full of them. I always think it's external signals, how much food have you got? Where are you located? How many buddies are around you? But we honestly have no idea.
I do want to point that out since you kind of put an emphasis on location. I really liked, and I should say we keep saying your talk. We should probably refer to the fact that we were at the CTEGD annual symposium where we heard you speak about your research. But what I liked was you had this kind of genetic component, but then you also had this kind of visual component that you were doing in parallel. Did you know from the start that was going to be pretty important for some of the questions, or was that kind of just like, oh, we should probably do both these things, and then came back and said, I'm glad we did both those things?
I'll be honest, it's the latter. Every time, and so we do a lot of animal research, which, we use mice for our infections because it's a real living host, which is, more relevant, but it also means that you are using animals. Mm-hmm. And I think that we should get the most out of every research animal that we use. In regards to that, I was like, well, maybe we'll need to look at this. And if we do, I would like to have the samples and not have to infect more mice. Mm-hmm, mm-hmm. And so it does mean that you end up with drawers full of stuff that you've never used. But it also means that if you do need to go back and look because something you see doesn't quite make sense and you need that visual context, you have it. And so we were really lucky that I'm a hoarder, I guess. And
a scientific hoarder.
Hoarder, not in my home life. That we could go back and look. And one thing that came from that is that reviewers do appreciate that you're not looking at a different animal. Everything comes from the same one. Mm-hmm. So the genetics, the imaging, the fluorescence imaging, all came from that same batch of animals. And so it's directly related, not, are you sure it happens every time? And that they're not just not connected to each other.
Yeah. I liked it because when the one time you had the hypothesis that maybe it was the parasites that were on the outside or on the perimeter that were getting a signal, but then when you actually looked...
I was wrong. I know, it was heartbreaking. I thought I was so clever. I just started the lab and I was like, yes, this is definitely going to work because it's so logical. And I love logic. And I was, I put all my heart into it and nope. Wrong.
But those things always lead to bigger and better things.
Yes. I never would have looked at the cell cycle and found that they were replicating had that first answer even been close to right. I would have kind of stopped. And so I guess maybe, what did they say? Adversity is the mother of... No, wait, innovation is...
I thought it was perspiration was the mother of invention.
Perspiration? Perseverance?
Perseverance.
I was thinking hard work. I was going with hard work. Physical labor.
Gotcha.
It's ideas. Like you have to, if something is challenging, you have to come up with a better idea.
Yeah. People are going to laugh at this because the whole time we're trying to talk it out, they already know the answer.
Yeah. Somebody's yelling, it's this,
I think that happens a lot on this podcast.
Yeah, that's probably true.
So one of the things you've brought up a couple times that I find really fascinating is the idea that bradyzoites inside this cyst, which has a cyst wall around it, they build a wall around themselves, can somehow sense what's going on beyond that. In some ways, it almost seems like it's a one-way door. They can take things in, but they can stay inside in stealth mode because the immune system apparently doesn't react to them. So how are they sent? Do they put sensors up on the cyst?
Oh, I wish I knew. Wall? That's so there's a couple of kind of ideas we have about this. We find these individual parasites that come out of the cyst, and I like to call them bloopers because they look like they're like bloop out the side. And we've gone through many different names for what they should be called because you can't write blooper in a paper or in a grant because people don't like that. But we think that maybe they are going out into the world to see what's happening. And based on that, it's not that within the cyst they can come back and tell them, what's going on. But their changes to the environment make gross changes to the brain that they're in. And so then it would be a case of, it's like a tide that comes and tells you, oh, the blood-brain barrier is leaky, or there's a ton of cytokines, or something is happening. And so it's not subtle anymore because of the change in the knowledge that one parasite has gained. And I think that within cyst, they can sense molecules that are coming across, which would be like nutrients, and there's transporters, and hopefully they have some idea what's going on outside. But I don't think that they can sense too much subtle stuff.
So is there any immune pressure on the cyst at all?
So it's funny, we sometimes see in slices of brain that there are cysts that have immune cells all around them, and it looks like they're dying. So I think that sometimes there is immune pressure and they do find these cysts. But I think that a lot of them, they're so well hidden behind that wall. And I think that that's why maintaining that wall is so important. I think that if they don't have a good wall, then their proteins accidentally get out into the world. And our immune system is like very, very good at finding things that shouldn't be there. And so we've tried to kind of count the number that the immune system can see, but it's pretty difficult because it seems like it's quite low. And I think it's an accident. I think that the cyst gets old and that wall is not maintained and it's not taken care of. And the parasites inside don't want that, but there's nothing they can do to fix it. Kind of, the wall is too damaged. A little bit of paint and plaster is not going to sort it out. And so sometimes they will leave and they'll egress and leave that cyst. And sometimes the immune system is faster and it catches them.
Right, because they are going to try to make new cysts. We should be very clear about
that.
Yes.
Yeah, the parasites have to be inside a cell. So if they are outside, their one goal is to find another cell that they can invade and set up shop in and make more of themselves.
On the topic of immunity, and I'm not usually the one to ask the immunological questions, but one way host cells protect themselves from invading pathogens is by presenting a portion of usually a protein from the invading pathogen up on their MHC molecules.
That would be peptides.
Yes, peptides. Yeah, thank you. And basically to say, hey, I found this inside me, and they're communicating to a T cell, and the T cell will look at it and says, yeah, that doesn't look good. Some parasites, Leishmania I'm thinking of, can shut down some of this presentation. Do we know anything about toxo? Does it affect presentation of these peptides?
I think toxo, so you can have vaccinations against toxo, so I don't think it can completely shut down any presentation of peptides that allows for immunity to develop. Some immunity is good, some immunity is not as good, not as effective. If you have toxo, you don't tend to get toxo again, but you have it forever, so that's not particularly helpful because you've still got toxo. So it does allow you to get some immunity. So it cannot prevent the presentation of those, but you don't get great immunity against toxo because of those two stages. And so the bradyzoites just hide. And there is some push to do vaccines that use both the acute stage and the bradyzoite stage peptides so that you can get immunity against both, because that's important. If we just use one stage and the other stage just hangs about, you've still got the disease.
So people have really been ignoring the bradyzoites in that kind of realm.
I wouldn't say ignoring. I'd say they're just a lower percentage. It's hard work doing something that takes a month to make the chronic infection. It's hard work to do it in mice. And it's hard when the lab adapted strain that most people use just doesn't make cysts anymore. So you have to do the less genetically tractable model if you want to do anything with cysts. And you can't look at anything that's important for the first stage because you need it to get to that second stage. And so our options are kind of reduced in what we can look at. And so I think when you don't know a lot about the parasite, you look at the stuff that's more accessible first. I think maybe I'm just a bit crazy for wanting to look at the thing that's the second stage and only happens in the animal, but some people's got to be crazy, right? Otherwise we never look at it.
Yeah. So when you're not trying to get cysts out of brains or muscle, I should say, actually, not brains, what do you do in your free time?
I love reading terrible, trashy novels. I read a lot of journals, and so I like to read mind-numbing books.
Get to
balance it out. I love cooking, and I'm a huge rugby fan. I used to play, but I had an eye surgery and they won't let me play anymore. So I just mostly watch it and yell at my TV or go to the games. And I absolutely love rugby. It is a wonderful sport that I recommend all Americans start watching.
I've actually watched a couple rugby games and I loved it, even though I had no idea what was going on. It's different. It's both similar enough and different enough to American football to be ultimately confusing. Because the ball will be loose on the ground and both teams are gathered around. And as an American, I'm thinking fumble. Whoever grabs it has possession. But they're not grabbing it. They're waiting, and then all of a sudden one of them will grab it.
And then the rest of them will pile on top of them to try and steal it away. Yeah. Yes, I think it's the violence that I like most about rugby.
Yeah, and they do all this heavy contact sport without helmets, without
pads. Yeah.
They wear mouth guards.
Okay.
Mouth guard is the one required thing for rugby to prevent concussions, or to try and prevent concussions when you're running around tackling each other.
Ouch. Well, our guest today has been Dr. Robyn Kent, assistant professor at the University of Oklahoma College of Medicine. Robyn, thanks for being with us.
Thank you. It's been wonderful.
Links to additional information about Dr. Kent's research can be found on our Instagram account, which is at PPPPodcastUGA.
As always, we appreciate you tuning in today. And if this is your first time listening to the People, Parasites, and Plagues podcast, we encourage you to check out the other exciting episodes exploring the research being conducted at UGA and beyond. Please rate the podcast, and if you have any questions or suggestions for future episodes, reach out to us via email at ppp@uga.edu. This podcast is brought to you by the Faculty of Infectious Diseases and the Grady College of Journalism at the University of Georgia. It is supported by the University of Georgia through the Office of Research, the UGA Graduate School, and the College of Veterinary Medicine. Special thanks to our production assistant, Justin Nemeth, for research, editing, scheduling, and keeping us on track.